Yeah, so we study a set of proteins called enzymes, and these are found throughout nature, and they're responsible for catalyzing chemical reactions that are necessary for life. And so what we really want to do and understand at a fundamental level is how these enzymes work, and also more importantly, how they evolve new functions so that we can leverage that for applications. So moving forward, I think the field of study that we're in is associated with a field called synthetic biology. And those synthetic biologists want to leverage proteins and enzymes to do useful things for humans, like bioremediate toxins or to design proteins to make scaffolds for a variety of different uses in biomedicine. And so the studies that we do really are trying to understand the fundamental principles by which proteins evolve their functions, and thereby hopefully being able to infer kind of general themes that will allow synthetic biologists to kind of move that field forward. Yeah, so we have two main projects in the lab right now, and they kind of can be divided up into looking backwards in evolutionary time to understand how proteins have evolved the functions they have. And then we have another project where we move forward in evolutionary time and try to challenge organisms to evolve new metabolic capacities. So in the first project, what we do is actually resurrect ancestral proteins. These are models of proteins that existed hundreds of millions of years ago. And we study the differences between those ancient proteins and modern proteins to try to understand how the function diverged and how the function evolved into those enzymes. In the later project, we use a process called adaptive laboratory evolution. And this is actually where we challenge an organism, a simple organism, to try to evolve new metabolic pathways or new metabolic capacities. And so this is especially important for things like, again, bioremediation of waste or trying to understand how organisms evolve things like resistance to antibiotics. So we use a lot of bacteriology and microbiology. Those are kind of the hosts for doing our adaptive laboratory evolution studies to develop new metabolic pathways. They're also kind of like our powerhouse for making proteins. So a lot of our work is producing recombinant proteins. So these are proteins that are maybe human in nature or ancestrally extinct that we produce in a bacterium, and then we kind of biophysically and structurally characterize those proteins. So I would say we do a lot of kinetic assays and a lot of biophysical assays, and a smaller part of our group is involved in X-ray crystallography and NMR structural determination.